In biochemistry, next one is organelles of the cell. So it's the first organelle is rough endoplasmic reticulum. There are two type of endoplasmic reticulum: rough endoplasmic reticulum and smooth endoplasmic reticulum. So what a rough endoplasmic reticulum do? It actually make all the secretory protein which are going to secrete and exported out of the cell. So first function is protection of the secretory proteins. Then the next one is it make proteins of endoplasmic reticulum, proteins of Golgi apparatus, protein used in lysosomes. So it make two kinds of protein: organelle protein and secretory proteins. The next function it also make peptide type of neurotransmitters in the CNS as Nissle's granules or Nissle's body, which is actually the endoplasmic reticulum of the CNS system. So three functions. The next one is it also rough endoplasmic also do glycosylation. Glycosylation is actually addition of glucose or to make a substance glycosylate. For example, the glycosylation or adding of the glycosylation any kind of sugar like oligosaccharides and make it glycosylate and a kind of protein. So in rough endoplasmic reticulum, actually the protein, a sugar mannose is actually glycosylate over the proteins, glycoprotein like acid maltase, and in this way it can transport it into the lysosome for further lysosomal functioning. So it add glycosylation or add sugar molecules. The next function is the organelles, which are the tissues which are rich in rough endoplasmic reticulum are the enterocytes, mucus secreting goblet cells of the enterocytes or the GIT, and also the antibody secreting plasma cells. So these are the main functions of endoplasmic reticulum. There is also free ribosomes which are scattered into the different into the cytosol and make proteins for mitochondria, make proteins for peroxisomes, and also make cytosolic proteins. And this cytosolic proteins, they are actually made by there is an enzyme known as not enzyme, there is a protein, cytosolic ribonucleoprotein, also known as signal recognition peptide, which is used to make signal recognition peptide. This is abundant in the cytosol, and these signal recognition peptide, they actually traffic or move all the polypeptide ribosome complex to the rough endoplasmic reticulum. So if there is abnormality in this signal recognition particle, then there is increased in the level of cytosolic protein in the cytoplasm. So the next is we have smooth endoplasmic reticulum. It is pretty much straightforward it do three type of work. It do steroid synthesis, it do detoxification, especially in the hepatocyte, detoxification of the poisons and the drugs, and it do G6 phosphatase enzyme, which is used in glycolysis and gluconeogenesis. Last step of this enzyme, last step of this pathway. And these are the pretty. So this smooth endoplasmic reticulum is the only organelle which do all the three kind of jobs: detoxification of amino acids, and production of the steroids, and production of glucose. So the tissues which are abundant in smooth endoplasmic rich tissues are actually the gonads, which produces different type of steroids, tissues which are hepatocytes, which detoxify protein and their drugs, and also glucose synthesis. So the adrenals, they use both steroid. So adrenals, hepatocytes, and gonads, these three tissues have the richest or the maximum percentage of smooth endoplasmic reticulum. The next thing is proteasome. So a body which degenerate proteins, degrade proteins. Which kind of protein? Two types of protein. If a protein is damaged, or if a protein is ubiquitination or ubiquitin tagged. And this ubiquitin tagging is very much important, because in Parkinson disease there is dysfunctional ubiquitin proteasome pathway, which leads to abnormal accumulation of aggregation of damaged proteins. Abnormal damaged proteins are aggregated in the brain, aggregated in CNS, causing this Parkinson's disease. So we have proteasome inhibitor. We have bortezomib, which is actually proteasome inhibitor. So how bortezomib helps us to treat multiple myeloma? In multiple myeloma, there are different type of spike protein, globulins, and these spike proteins are actually causing degenerative neuronal symptoms. So we use proteasomes to inhibit the functioning of proteasomes, so the cells, they naturally follow the course of apoptosis. So all the proteins, they naturally follow this apoptosis process. So this is the mechanism of action of bortezomib. There is also a drug known as lenalidomide, and it increases the E3 ubiquitin. It increases the E3 ubiquitination ligase, and by this ligase enzyme, to increase tagging of different proteins, which leads to treat this multiple myeloma condition. So there is increased tagging, increased ubiquitination, so increase protein degradation. The next is peroxisomes. So for peroxisomes, these organelles are pretty much important in oxidation process. So beta oxidation of very long chain fatty acid, and alpha oxidations of branch chain fatty acid. So these are the oxidizers of fatty acids. The next thing, they catabolize amino acids and ethanol. Also, peroxisomes, they synthesize bile acids and cholesterol. And the last is they also synthesize a compound known as plasminogen, which is actually CNS white matter membrane phospholipids. So these are the basic functions of peroxisome. So if these functions are not working properly, then we have a condition known as Zellweger disease, which is autosomal recessive type of problem, in which there is a PEX or peroxisome gene abnormality, which leads to deficient enzyme used for oxidation. So no biogenesis, no beta oxidation. So the proteins, the fatty acids which are not oxidized, they are going to aggregate in the body, causes hypotonia, seizures, and all CNS abnormalities. Also, they can cause hepatomegaly. So these two are very much important signs to diagnose a patient suffering from Zellweger disease. So this is Zellweger problem. Then we have the Adrenoleukodystrophy, which is an X-linked recessive problem, in which also there is a problem a mutation of ABCD1 gene, which can lead to no biogenesis or no beta oxidation of the branch of the very long chain fatty acid. So the precursors are aggregated in the body, and they are aggregate in adrenals, causes adrenal crisis. They are aggregated in the leuco or white matter of the brain, CNS white matter, causes the progressive neuro losses, and also they have problems in the testicles. So the most important symptoms are neurodegeneration and adrenal crisis. And the last one is Refsum disease. It is autosomal recessive problem. It is pretty much new, not very much tested in the U world. So Refsum problem there is abnormality relating to branch chain fatty acid oxidation abnormality, which can lead to aggregation in the body, same mechanism, and it has pretty common symptoms like scaly skin and ataxia. Then we have cataract, night blindness, then the bone problem, epiphyseal dysplasia, ataxia, and short fourth toe. So how to treat? We change the diet and plasma pheresis. So these are the pretty much important organelle